A very good day to Dr. Nikman, Dr. Vijayan and everyone. We are Group K from SIO1003 Bioinformatics Concepts and I am Sanjana Sri Chandrasekharan, the group representative. Today we will be presenting on CRISPR-Cas9 gene editing, focusing on bioinformatics tools for guide RNA design and off-target prediction and how these tools improve accuracy and safety. These are the list of group members from Group K.
Today I am going to talk about CRISPR or Cas9 gene editing. This technology has completely changed how we approach genetics and medicine, and it is honestly one of the most exciting breakthroughs in science. Let's start with the basics. Tiny organisms like bacteria and archaea are constantly attacked by a virus called bacteriophages. To survive, they develop a smart defense system called CRISPR, which stands for clustered regularly interspaced short palindromic repeats. Here what happens? CRISPR stores little piece of DNA from viruses that attacks before, kind of like a memory bank. When the same virus comes back, CRISPR recognizes it and with the help of a protein called Cas9 cuts up the viral DNA to stop the infection. Scientists saw this and thought, if bacteria can do this, maybe we can use it to edit genes. And that's how CRISPR became a powerful tool for gene editing. It's faster, simpler, precise than the older method. What is CRISPR? Think of CRISPR like a pair of molecular scissors guided by a GPS. The GPS is the guide RNA and the scissors are Cas9. Together they find the exact spot in the DNA and make a cut. This allows scientists to remove, add or change the genes. The process happens in 3 steps: memory acquisition, storing viral DNA fragments as a defense memory, guide the RNA formation, like creating that tells Cas9 where to go, and then cutting the gene. Why is it important? CRISPR is a game changer because it let us edit genes with incredible precision. It speeds up the biomedical research so we can understand disease better, and it has huge potential for treating genetic disorder, even cancer in the future. Pretty amazing, right? Why do we need bioinformatics? Now, here's the catch. CRISPR isn't perfect. Sometimes Cas9 cuts the wrong spot in the DNA. These mistakes are called off-target effects and they can cause harmful mutation. That's where bioinformatics comes in. It uses computer tools to predict where these mistakes might happen, design better guide RNA that are super accurate, and make sure everything is safe before we use it in real patient. So bioinformatics makes CRISPR safer and more reliable. To wrap up, CRISPR is one of the most exciting breakthroughs in science. With the help of bioinformatics, we are making it safer and more precise, and that means a future where we can treat genetic diseases and maybe even cure them.
Now I will talk about the CRISPR design workflow. This slide shows the basic steps of how CRISPR-Cas9 is designed. First, the target gene is identified and located in the genome. Researchers need to clearly know which gene they want to edit. Next, we identify the PAM sequence near the target gene. A PAM stands for Proto-Spacer Adjacent Motif, and is required for Cas9 to bind and cut the DNA. After that, the sgRNA is designed. The sgRNA must be complementary to the target DNA sequence so that it can get Cas9 to correct location. Then, the sgRNA is tested on both the target gene and possible off-target genes. This step is important to check whether the sgRNA is efficient and specific. Finally, after screening, the best sgRNA is selected and combined with Cas9 to form a RNP complex. This complex will be used for gene editing experiments. Okay, the next slide, I will briefly introduce the on-target criteria for sgRNA design. First, the sgRNA sequence must be complementary to the target sequence to ensure correct binding. Second, repeated sequences should be avoided. This is because repeated sequences can form secondary structures, such as hairpins, which reduce efficiency. third, the prime sequence must be located next to the target genes. Another important factor is GC content. The GC percentage should be between 40% and 60% for optimal sgRNA activity. Finally, the target sequence should be unique in the genome. This helps reduce binding to off-target site and improves specificity. This criteria helps ensure that the CRISPR system works efficiently and accurately. And with the proper design and on-target criteria, we can improve the success of criteria gene editing.
Hi, I'm Lin Weijia. To begin our look at the challenges of gene editing, we must address off-target effects, which occur when CRISPR accidentally cuts a DNA sequence that is wrong but very similar to our intended target. This happens because the gRNA binds to a sequence that almost matches the target, leading the Cas9 scissors to make an unintended cut. There are four primary reasons this mistake occurs. First, Cas9 is somewhat flexible and may still cut even if 1 to 3 letters of the DNA code do not match the gRNA. Second, the human genome is so vast that look-alike sequences often appear by chance in different regions. Third, a poorly designed gRNA might lack specificity, meaning it could naturally match up to 20 different spots in the genome. Finally, our DNA is personally unique. S and P, which are one-letter differences between individuals that can create unexpected near matches for the CRISPR system to target. Because safety is a priority in bioinformatics, we use a three-step approach to identify these risks before they become a problem. We start with in silico methods, which are computer-based, such as using software like CRISPR or Cas9 to scan the genome and predict where off-target cuts might occur. Once we have these predictions, we move to in vitro testing in a test tube environment, where we mix Cas9 and gRNA with purified DNA. A common example of this is digenome sequence, which shows us every physical cut the system makes. Finally, we verify this result in vivo, meaning inside real, living cells. By using advanced sequencing methods like guide sequence or circle sequence, we can identify actual off-target cuts as they happen in a live cellular environment. By combining these computational and experimental strategies, we ensure the highest level of precision in our CRISPR designs. That's all from me. Now that we understand the risks and how to find them, I will hand it over to our next presenter to discuss the major tools we use for these searches. Thank you.
Hi, my name is Wijaya, matrix number 24212096. I am presenter 4 and I will be presenting slide 10 and 11. I'm gonna begin with slide 10, which shows the key tools for CRISPR design. The first tool is CRISPR. CRISPR predicts both on target efficiency and off-target sites, which tell us how well and how safely a guide RNA will cut a gene. It also provides guide RNA scores and genome specificity information, which makes it checks how unique the target sequence is in the genome. In addition, CRISPR integrates with multiple genomes, allowing researchers to design sgRNAs for different organisms, such as humans, plants, animals. And the second tool is ChopChop. It's a user-friendly web tool for designing sgRNAs. It allows researchers to visualize the target gene so they can see exactly where the CRISPR will cut. It also provides filters for PAM sites, GC content, and off-target risk that can help scientists guide RNAs that are both stable and accurate. The third tool is Benchling. It's a cloud-based platform that combines CRISPR design with lab management. So scientists can design sgRNA, plasmids, and DNA sequences in one place. And it also supports collaboration, meaning multiple researchers can share data, comment on designs, also track experiments together. This makes CRISPR research more organized and more efficient. Now I will move on to slide 11, which shows a case study using CRISPR. The target gene in this example is BRCA1, a gene involved in DNA repair and linked to breast cancer. The objective is to select an sgRNA that can target this gene accurately with minimal risk. So the first step is entering the BRCA1 gene sequence into CRISPR. Then CRISPR generates several possible sgRNAs and will give each one an on-target efficiency score and an off-target prediction that can tell us how strong and how safe each guide RNA is. Step 2, there will be 3 sgRNAs that are compared. sgRNA 1 has 75% efficiency and only one off-target, meaning it is very specific. sgRNA 2 has 60% efficiency and 2 off-targets, so it is safer but weaker. sgRNA 3 has the highest efficiency at 82%, but it also has 4 off-targets, which makes it more risky. So step 3, the decision is, although sgRNA 3 is the strongest, its high number of off-targets increases the chances of unwanted DNA damage. So the best choice is sgRNA 1 because it provides the best balance between efficiency and accuracy. So this case study shows how CRISPR helps researchers compare different sgRNAs and select the most reliable one for gene editing.
Okay, so now we move on to the applications of CRISPR and Cas9. So the first application is in the medical applications, in which in gene therapy we can use gene editing to correct mutations-bearing diseases, and also we can use it in infectious diseases and cancer treatment, in which we can edit immune cells to properly target, recognize, and eliminate cancer cells and viral genomes like HIV and HPV. And then in agriculture, we can use CRISPR to target specific genes that were associated with desirable traits that we want, like disease resistance, pest resistance, and drought tolerance. And this can overall improve crop quality. And then in research and in biotechnology, we can use it to improve our genomics understanding by using the enumeration concepts of knockouts, and that will enable us to create modern organisms faster and more efficient than conventional methods. And then we can also use CRISPR to create more complexity, to build more sophisticated engineering of biology, like enzymes production and also biofuels. And so that is it. That is the overall usage of applications and interest.
To sum up, the CRISPR-Cas9 is a modern tool used to change genes. It uses a small guide RNA to show the Cas9 enzyme exactly where to cut the DNA. After the cut, the gene can be fixed or edited. Sometimes, CRISPR can cut a similar place by mistake, and this is called as an off-target defect. To avoid this, bioinformatics tools such as CRISPR, ChopChop, and Benchling help check the DNA and choose safer guides. Because of this, the applications of CRISPR is becoming very important in health, agriculture, and also scientific researches.